Biology and Big Data. This is Biostatistics Core Special Topics Talk number 17, an overview of bioinformatics. I'm Mark Williamson for the DaCCoTA at the University of North Dakota. Let's get crack-a-lackin'. So, the goal of this talk is to give an overview, a very broad and slightly non-canonical overview of bioinformatics. So what we'll be talking about today, some definition and history, then moving along to field and topics of bioinformatics, then types of bioinformatic data, methods, and machines and software used in bioinformatics, moving on to common databases, some example applications, and finally round it out with the future of bioinformatics. So, getting started with definition and history, Merriam-Webster defines bioinformatics as the collection, classification, storage, and analysis of biochemical and biological information using computers, especially as applied to molecular genetics and genomics. So if you think about the word bioinformatics, two parts, bio, life, informatics, so information. So this is the study and use and modification, handling, whatever you want to say, of biological information. So it's mainly through the use of sequencing data, so DNA, RNA, proteins, and the downstream applications therein. So a great little history here I found from a publication on PubMed from NCBI. So this is reference number 1 here. Goes through the history of bioinformatics. So in the 60s and 70s, it really actually started not with what's most commonly used today, which is DNA. It started with protein analysis. That was its origins. And one of the big founders of this was Margaret Dayhoff, which she was the first real bioinformatician. And so here's an example of what some imaging using very rudimentary computers, punch cards, etc. And actually, it turns out the first bioinformatics software is called CompProtein, and it was used to try to align proteins. Next, the 70s to 80s, there's a shift from protein analysis to DNA analysis. And this is where a very important algorithm that's still being used today, as an example, the Needleman-Wunsch global alignment algorithm, where you're trying to align DNA sequences, very common task in bioinformatics. As the 80s came into the 90s, there were certainly advances in biology and computing, so more could be done. In the 1990s and 2000s, that's where genomics, so sequencing whole genomes, really came into the light, and the start of using bioinformatics on the internet. Early 2000s to 2010, that's where high throughput bioinformatics came online. This was also known as second gen sequencing, where you could actually do higher level bioinformatics because the technology was catching up, and also the computation. And finally, since about 2010 to today, a lot of what bioinformatics has been starting to deal with is certainly the advent of big data, not only in bioinformatics, so biological sequences, but data in general, the shifting role of bioinformaticians, and as well as tackling bigger topics that, like system biology, that couldn't really be dealt with until we had the computational power. With that, let's move on to some fields and topics in bioinformatics. It's such a sort of widely applicable field, or there's a lot of moving parts, so it's really hard to really pin down exactly what sort of fields bioinformatics covers, because it touches a lot of different fields. So I found this is the best infographic I could kind of find, and you can see bioinformatics covers a lot of things from biology, biochemistry, chemistry, computer science, engineering, mathematics, statistics. You see it kind of touches all those topics. So an important discipline that I felt was left out here was actually medicine, so I sort of wedged it in there. And what sort of topics it covers, or maybe you could call applications from the same reference, you can see that really a lot of times starts with DNA sequences, goes to bigger, so genomes. You can look at evolutionary biology. That now that you have genetic sequences, now a very strange sort of niche thing that this infographic brought up here is immunoinformatics, but certainly something that can be done. Gene expression, proteomics, so that's certainly common topics. Though, again, these are pretty simplified views. I couldn't really find a great overarching infographic of bioinformatics, so I did my best to kind of make my own here. This is taking topics and subtopics from the Wikipedia entry on bioinformatics and trying to piece it out here. So the big picture here's one, subtopics are in this orange. So from sequence analysis, gene and protein expression, etc. And then these lighter circular ones are subtopics from there, trying to branch and connect it the best I can find. And so this kind of tries to give a broader view of what you can do in bioinformatics, from anywhere from DNA sequencing to working with network analysis with databases. And there should be a link in the description below on, if you want to just, because obviously I'm just showing you this infographic and not going to dig into it here, but there should be a link to, if you want a PDF of this infographic to, say, post on a wall like someone might have done, namely me, if this helps you in any sense of trying to get a handle on bioinformatics. Because again, it's sort of a very wide field. There's a lot to be done. So it's hard to really capture it in one snapshot. This is my best attempt here. Anyways, let's move on. We've wasted enough time in the fields and topics. Let's drive along to the data. So the fundamental data that this comes from is sequencing of biological sources, taking the source code, the DNA, so those nucleotides A, C, T's, and G's that pattern across sequences, and getting that into computer format, getting that into information. And of course, there's also information on RNA and proteins. So again, on the DNA level, it can be just sequences, so bits of DNA. It can be whole genes. It can be whole genomes, so all the DNA of an organism. It can actually be metagenomes, so organisms from a bunch of different species in say, a sample. Against RNA and proteins. And then there certainly can be others. Here's a reference that shows some other ones. Some examples is often data will need metadata with it. So like location, sample, species, whatever. You can actually do bioinformatics with images. Images are very data rich, and so you can do things with that, etc. You can see this resource for more examples. So that's sort of where most of the data comes from, these biological sources that are sequenced by some sort of machine that we'll see later, and then put into some sort of format on a computer to be analyzed. So very common file formats of bioinformatics data. The two big ones are FASTA and FASTQ. So FASTA files, I'll give you an example here. It's the DNA sequences with some sort of header. So the header will have this top here. It can be very complex. So this has a lot of information on the species, others sort of information, and then followed by a line with all the A, C's, T's, and G's. They can also be much simpler. It could just be like, it could be something like sample 1, sample 2. And then the difference between a FASTA and FASTQ, a FASTQ has a couple additional lines, including the Q is quality. It'll actually have a quality score for all the different sequence. So you can see how well, because whenever you sequence things, there's a certain efficiency or well-knownness about it, a certain accuracy, and so quality scores tells you how much you can trust each sequence. So maybe these first couple ones aren't very reliable at all, but if you get later on to the line, it has very high quality, so you can really trust that that is the real sequence. There's not ambiguity. So then there another common type of file, because often what you're doing is you're getting multiple sequences and you're trying to line them up. So there's alignment files. There could be BAM, SAM, or CRAM files. And then there's all sorts of other files, like VCF, which is variant calling format. It stores gene sequence variations, such as SNPs, and is used for genotyping projects. GFFs are generic feature formats, describes the various sequence elements that make up a gene, and it's a standard way of annotating genomes. There's DTF, gene transfer format. It's like GFF, but it's used to define the gene and transcript-related features exclusively. BED files are browser extensive formats. They're used for visualization in a gene browser. tar.gz files, those are compressed file types, so often these other types of files will be compressed with a tar.gz, and you need to unzip them or uncompress them. PDB is atomic coordinates for storing 3D protein structure by the Protein Data Bank. PED files are for pedigree analysis. Map files accompany PED files. CSVs are comma-separated values. These text files can be opened in Excel, so it's very useful to be able to examine these types of files, and these often will be metadata files or others. And then there's JSON JavaScript object notation. These are common in other industries, but apparently they've been growing quite a bit in bioinformatics, so we'll probably be seeing them more and more. With that, let's talk a little bit about methods. This is not going to be a completely exclusive setup, but hopefully give a good overview. So first of all, I want to talk about pipelines, because a lot of what goes on in bioinformatics is not just a single step. It's a bunch of different steps chained together. So you take data in one form, you do something to it, that modified data goes to the next step, to the next step, to the next step. So that's very common to have pipelines in bioinformatics. So as an example, I took these examples sort of generic format based on this reference from a Coursera course on whole genome sequencing and kind of adapted for this example. So usually any sort of bioinformatics project, you'll start with some sort of research question you want to answer. You'll design your study. You'll collect and handle your samples. So it's DNA, RNA, whatever type of biological origin it comes from. You need to make sure you store it right, etc. You're going to extract the DNA or RNA or whatever. You're going to prep the library, and you're going to sequence that data using some sort of machine, some sort of process to get it into a computer file. And then you're going to analyze that, work on that, maybe do statistical analysis, and then have some sort of results. You want to report it. You might need to store your data and share your data with the world, because hopefully your bioinformatics project is interesting and solves fascinating questions, and you can't wait to tell the world about your findings. So with that, let's talk about probably the baseline method. Usually the most common thing that you start with is DNA sequencing. And of course, that will have a lot of different steps. Now, of course, there's the wet lab stuff where you actually have to extract DNA, spin it down, break it up, filter it, add primers, PCR, blah, 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 throw it into a machine, get the sequences out. That's certainly steps, but there's also steps once you get it into essentially code. You'll need to do things like trim, check for quality, filter out poor quality things. You might need to, if you're assembling a genome, you might need to put fragments together. So there's a lot of steps that will happen, and what you're trying to do or what platform you're using, those steps might be different. Now, on the base level, there's two main types of DNA sequencing. One is amplicon sequencing, where you're just taking a certain segment of a genome. So, and usually with amplicon sequencing, you're sequencing a lot of different samples. The most common here is 16S or 18S. That's good for determining, like, say, who's in a sample, like what microorganisms or whatnot are in a sample by the 16S ribosomal RNA gene. Also, there's other things you can do, like other single copy genes or other genes of interest, but that's really all going to be the same general process. You have a gene or part of a gene that you're sequencing across samples. Whereas the other main type of DNA sequencing is genome sequencing. You're doing the whole genome of a species and maybe multiple species at once. The two subtypes would be reference-based. So if you already have a genome, the genome's already been sequenced, you can essentially use that as a reference and build your own genome. So a great example would be the human genome has been sequenced, but maybe you want to sequence your own genome. You sequence that, and then you compare it to the reference genome, and then can build that up. But if you don't have a genome sequence, and every genome at one point didn't have a genome sequence, you have to do it what's called de novo. So from scratch, you essentially have to assemble it without any sort of reference, which is certainly a difficult problem, but there are methods for that. And then another thing, jumping off from DNA sequencing, is typical next step would be annotation. So what's there, what genes are there, what might they be doing, what types of information in the genome is going on. Then another big thing there would be comparison, because usually with a genome or amplicon or whatnot, you might want to compare to what's different between samples or different organisms. That's a big part of bioinformatics, is comparing genetic information across categories, whether it's different species, different subspecies, different samples, whatever. Then beyond DNA sequencing, there's of course gene and protein expression. Gene expression is typically done through looking at RNA, because DNA is the gene, so it shows you what's there, but just because it's there doesn't mean it's actually making anything. So messenger RNA, looking at the expression of messenger RNA will tell you how well a gene is being expressed. And then ultimately, though, messenger RNA gets transcribed or translated into protein. And then protein are actually what does things in a cell, in an organism. So it's often really useful to look at protein expression as well. And proteins are big, complicated, and have a lot of interesting challenges to solve as well. Some common methods within this arena is like RNA-seq, also microarrays. Then beyond, once you have DNA, gene, whatever, you can also do phylogeny. So that's looking at evolutionary history, like how well are organisms related to each other based on their DNA sequences or protein sequences. And then, of course, you can do network analysis. That's another higher level, more complicated way of looking at things. And then more broadly, bioinformatics deals with any sort of omics data. So omics is sort of tacked on to any sort of biological thing here, which deals with large data. So genomics is the study of the genome, genomes. You could have proteomics, study of proteins, transcriptomics, that's transcribed RNA. But there's all sorts of omics you can think about: metabolomics, culturomics. I've even heard things like foodomics or interactomics, immuno-omics. So that the sky's the limit when it comes to what you can do with omics methods. All right, moving on now. Let's talk a little bit more about the machine. So how do we get from the biological material to information on a computer? So the biggest thing I'll cover here is DNA sequencing. There are 4 main platforms, so I'll kind of go through them. There's some older platforms that are no longer in service, but I'll just cover the main ones that are currently operating. So there is Illumina, and these are a short way of short read, and it's probably the most common. So essentially, genomic DNA is taken, it's fragmented, you add adapters, and then there's this thing called a flow cell, which these fragments will attach to, and they'll have what's called bridge PCR. They'll be amplified. Essentially, as these clusters are made and nucleotides are come and added on, the nucleotides have different colors based on whether they're A, C, T, or G, and they'll flash a color. And so the computer will actually look down at these cells, at these colors, and record the colors, and then that will tell what the sequence is as it's being built. And then so at the end of it, you have a bunch of these different fragments with the sequences, and then from there, if you're doing assembling a genome, you try to put all these fragments together. Next, there's Ion Turret, which is also short read and bridge PCR, though the main difference here is rather than on sort of a flow cell, this flat surface, this happens on magnetic beads. Oxford Nanopore is very interesting, and we'll see in a little bit with some pictures, it's actually a really portable technology. It's very cool, but it does something very differently. And so how it sequences is there's actually this protein pore. The DNA comes and it's unraveled, and as it's unraveled and goes through the chain, there's a measurement of the change in current, and depending on the current, it'll read out the sequence of A's, C's, T's, and G's. And finally, there's specific biosystems, which is really focused on long reads. So something like Illumina has fairly short reads. That's the limitation of the technology. And there's problems with short reads and assembling long things here. So long reads are really nice if you can do it. However, one of the downsides to something like specific biosystems is the longer reads it's a lot more expensive to run these, and in fact the sequencing errors, so the accuracy is a lot lower. However, as technology gets better, the cost is coming down as well, and the accuracy is going up. So long read technology is certainly going to become more and more important as time progresses. So that was some big things on DNA sequencing. Let's just take a look at some example of what the machines actually look like. So here's Illumina, Ion Torrent. Here's Nanopore. You see that it's actually really small and portable. It's almost like it can be as small as essentially a cell phone and plugged by like a USB, powered by USB. So you can really bring these anywhere. They're really cool. I really have only worked with Illumina, but Nanopore just makes me excited. And then finally we have Pacific Biosystems. And of course there's different machines for different processes within these companies. So multiple machines for different uses. But that's some general pictures. Okay, next let's move on. Of course, I can't cover all the sorts of machines that could be used in bioinformatics, but I'll just cover some other top ones. So there's mass spectrometry. This is a way to measure the mass to charge ratio of ions. So it's really great for protein identification. And there's lots of different types of mass spectrometry. But that's one way you can determine what a protein is made of or what some sort of chemical is made of. Another thing that's often very useful is flow cytometry. It's a technique used to detect and measure physical and chemical characteristics of populations of cells or particles. So you can stain cells with different staining for different properties, then run them and actually for each cell have an information on them. So that's pretty powerful. All right, with that, let's talk a little bit about software, because the machines were sort of the biology to computer side. So once we have the information in a computer, what can we do with it? So there's so much software. It's proprietary, open source, different methods. So I really can't go through all of it here, but I'll try to kind of cover conceptual categories. So I'll start with some core tools. So there's lots of different tools. Probably one of the most common one is BLAST. So this is basic local alignment search tool. So it finds regions of similarity between sequences. So if you have a sequence that's unknown, you can essentially compare it to a giant database of sequences that have identification, and you can see which one it closely aligns to. So there's a lot of different flavors. You can look at nucleotides, or proteins, so a lot of things you can do here. There's things like MBOSS or Cluster Omega. Cluster Omega is a multiple sequence alignment program. There's Prospect, there's Ensemble. This is a genome browser for retrieval of genomic information. There's USearch down here. It's a sequence analysis tools. There's SAM tools, set of utilities for interacting with and post-processing short DNA sequence read alignments. There's things like Velvet, a de novo genome assembly, Bowtie, sequence alignment and sequence analysis, Top Hat, throughput alignment of shotgun cDNA sequence reads using Bowtie first. Muscle is another one of these multiple alignment software. There's Protein Tools, which is an open source toolkit to analyze protein structure, and the list goes on and on. Other things maybe to think about conceptually, there's a lot of these have underlying code, sort of algorithms that are used. I've already talked a little bit about the Needleman-Wunsch. There's also the Burroughs-Wheeler's transform. Both of these are really helpful for alignment and other things, because you're almost always starting with some sort of sequence alignment. When you're getting into things like making phylogenetic trees, which is a very common problem to try to solve, there are common algorithms like the UPGMA, the unweighted pair group method with arithmetic means. That creates rooted trees, whereas something like neighbor joining is a general clustering and tree creating one that makes unrooted trees. And again, there's more underlying code, but I thought these are some of the most well-known ones that you might hear, so it might be good to have a little bit of a description. Now, next I want to talk about software environments. These aren't so much specific software, but whole environments used to support bioinformatics. So first of all, there's BioPython. These are a bunch of packages, tools, libraries to use bioinformatics with Python. And I've used this a little bit here. There's also Bioconductor. This is open source software for R packages. So if you work in R, Bioconductor is your jam. And of course, any sort of computer language will have probably something like BioPearl, etc., etc. Next here, again, we're going to go into that pipelines things. I already talked about this earlier, but that's a very important thing. There'll be set up specific sets of code or sets of programs to fulfill a certain bioinformatics pipeline. So some common ones are like data 2, DNA. That might be more specific, like DNA-seq analysis. Here's the example of the pipeline from that. You might have, for example, the Center for Genomic Epidemiology. They have pipelines for antimicrobial resistance gene finders. You might have things like CHIME2, Mother. These are common pipelines. And then more broadly, sort of even at a higher level, you can even have things like workflow managers, where you're not just having a pipeline, but also working the workflow and how you connect to, say, databases or servers. And so this is or even just setting up pipelines in an environment. So this is an example of sort of a general overview of what a workflow manager might look like. And a very common, popular workflow manager is the Galaxy project. Okay, let's talk a little bit about databases, because one of the big things with bioinformatics, it creates a lot of data, and then that data needs to be stored so it can be used, referenced in all sorts of other bioinformatics projects. So first of all, it's just some ideas on some of the big entities that house databases. So first of all, in the US, there's the NCBI, the National Center for Biotechnology Information, that has a lot of database references. There's the European version of that is the EMBL, and then also there's the Japanese Institute of Genetics. So those are probably the big three. If you think of entities that have bioinformatics databases, those will come up. So for things like DNA, common databases are places like GenBank, RefSeq. For proteins, it might be databases like UniProt or AlphaFold. AlphaFold is really cool because it has protein structure predictions using AI. For pathway analysis, the most well-known of these are KEGG, the Kyoto Encyclopedia of Genes and Genomes. For things like taxonomy, you have NCBI taxonomy, catalog of life, etc. And now some really interesting databases that I've either used or I'm interested in using for specific applications of bioinformatics. So I thought I might share them with you. One is the RRNDB, the ribosomal RNA database, which talks about ribosomal RNA copy number, which is differential in organisms. This is specifically for prokaryotes, so that's pretty interesting. There's the Genome Taxonomy Database, the GTTB, the most sort of comprehensive and systematic nomenclature for prokaryotic taxonomy. So it's pretty cool there. I keep seeing that pop up in papers I read. And then finally, another really interesting database is Silva. These have high quality ribosomal RNA ribosomal RNA information on sequences. So it's a database for that. Okay, moving on, let's talk about just some example applications. Now, there's all sorts of things you can do, from basic sequencing to genomics, but these are some interesting applications I think I'd like to just bring up here. So first of all, there's metagenomics. So this is looking at not just the genomes of one organism, but all the organisms in, say, a sample. And this has really revolutionized what we know about the microorganism, the microuniverse, really. That because a lot of microorganisms in, say, soil or water or wherever could be cultured, so we couldn't detect them through normal means. But we could sequence their genomes, and we found that, oh, wow, there's a lot more diverse organisms in there, bacteria, archaea, etc., than we could even understand. And so this is a really exciting field. And of course, there's a lot of complications because you're not just trying to get one genome out, you're getting a bunch of genomes out, sequencing, trying to assemble them and separate them from each other. And you can answer really cool questions like who's there, what can they do, in fact, how are they different between organisms or between samples. Another useful application is systems biology. So this is really trying to connect different levels in biology, not just sort of the traditional approach to biology would be sort of compartmentalize or sort of cut things down to one little area. Like you just study one part of a cell, but of course a cell, say a ribosomal RNA in a cell isn't working on its own. It's in a system of all sorts of other components, and then there's higher levels. There's organelles, there's cells, there's organisms, and so systems biology tries to sort of synthesize across these different systems the answers to these holistic big picture questions. So that's a really computationally complicated question. So bioinformatics certainly can help in this field. Next is sort of a interesting thought of an application is actually literature analysis. Even just thinking about this for biomedical research, there are millions of papers in, say, cancer, and you can't just, if you're looking to try to search through these, you can't do that all by hand. So the idea of being able to sort through papers and pull out important information and organize, cluster, etc., all this vast literature has a lot of use for using bioinformatic type tools to deal with those problems. So that's an interesting sort of dark horse application that you might not think about immediately if you're in the like DNA, RNA sort of biological frame set. Then finally, I want to talk about microbiome research. This is a really exciting field because this is the study of all the microorganisms in some sort of biome, some sort of area, and especially the human microbiome. So looking at the collection of bacteria, etc., in the human body has been really exciting, especially like the gut microbiome. We've learned a whole lot about that and sort of upended some of our thoughts about how biology works. And so for example, this is a picture from a paper that looked at microbiome studies, human microbiome studies of cancer cells across cancer cell types. So there's a lot of really exciting frontiers in microbiome research. And of course, these aren't all mutually exclusive. Metagenomics essentially is, microbiome work is essentially a subset of metagenomics. So it's not like these are mutually exclusive. Okay, so with that, let's talk a little bit about the future of bioinformatics. So a big thing with bioinformatics is growing every year. There's an increasing market share as time goes on, as we get better sequencing tools, more data, more need to analyze data to answer complex questions. So sectors of bioinformatics are growing, applications are growing, technologies and services are growing. And as a sort of a basic overview or a basic post from LinkedIn, reference 32 here, talked a little bit about with bioinformatics, there's going to certainly be a growing role of robotics. So that's more maybe on the automated DNA sequencing side, that sort of wet lab stuff, but also growth in things like machine learning, deep learning, AI, etc. Of course, AI is all in the news today as a time of this writing, so that's just going to continue to be used, that type of those tools for bioinformatic processing. Finally, I mean, really, there's going to be more data, more integration, so there's going to be a need for more tools to deal with the growing bioinformatics infrastructure. And I guess one thing I want to note of importance, I've talked about this a little bit with the Pacific Biosystems, is long read technology. Ben Langmead, in one of his great talks on, or series of talks on algorithms for DNA sequencing, which we'll see in a little bit, he talks about the future being long, where the use of long read DNA sequences, which solves a lot of problems of trying to assemble genomes. There's a lot of problems with having short reads and trying to map them and set them up. That's a whole unsolved, tricky problem, which can be offset a lot by these long reads. So it's certainly the case that long read DNA sequencing technology, so at the baseline, is going to really probably be the future, and that we'll see that more and more in bioinformatics applications, which will hopefully solve a lot of these intractable problems or really tricky problems with short read technology. All right, with that, I want to go through some resources for you to use. This is, of course, it's a very broad overview, a very scattered overview. I am no whip-smart expert in all manner of bioinformatics. I am trying to learn more myself, so these are some resources that I found helpful or I'm looking into that hopefully will help you get to the next step if you want it. So I talked about Ben Langmead. He has a great talk on algorithms for DNA sequencing. You can find these on YouTube. And so this really gets into some of the code or algorithms, what's going on underneath the hood of a lot of DNA sequencing, DNA alignment. It's a fascinating set of series of talks. I cannot recommend them highly enough. Next, the Joint Genome Institute has a lot of great information here, including several YouTube playlists on conference talks. They had this New Lineages of Life 2021 symposium, which if you want to know anything about Archaea or New Lineage of Life, I was able to go through this series. This blew my mind. It was fascinating. Again, can't highly recommend it enough. They certainly have other great talks that I hope to get to and may be interesting, like the Vega Symposium or the Annual Genomics and Energy and Environmental Meeting. Next, there's the Center for Genomic Epidemiology. That's from the Technical University of Denmark, TDU. They have a couple free talks in Corsia that I've took that are a helpful introduction to some bioinformatics. One is metagenomics applied to surveillance of pathogens and antimicrobial resistance, so a little more specific application. But then a broader one, which is whole genome sequencing of bacterial genomes, tools and applications. So those are a great start if you want to get more into bioinformatics, especially looking at like sort of a pipeline of how you go from one thing to another. And finally, I found a great resource, bioinformatics.ca Canada. It has a lot of great student workshops. Just in 2021 alone, these were some of the workshops: pathway and network analysis, machine learning, cancer analysis, introduction to R, analysis using R, microbiome analysis, RNA-seq analysis, epigenomics analysis, high-throughput genome analysis, infectious disease epidemiology analysis. So a lot of great resources you certainly can dig into there. With that, let's go through my references. Of course, these should be in the description to this video below, but I want to be a good scientist and show my sources. So we'll have pages of references here. And again, throughout the talk, when you have the number in the brackets, it refers to these reference links here, both pictures and site. And with that, I want to acknowledge the DaCCoTA, the DaCCoTA Community Collaborator on Translation Activity. That's what supports me. Most of my support comes from this. So if you found this talk really useful, please acknowledge the DaCCoTA for publications. So hope this was a helpful overview, however skimming. Now go forth and grow your bioinformatics bona fides. Thanks for joining.